Chapter 24, Urinary Physiology, Screencast 4. This is the fourth and final screencast for Chapter 24. The previous screencasts have focused on the production of urine and kidney function. This screencast will actually be focusing on urine and the elimination of urine. Urine is a product of filtered and processed blood plasma. Generally, we consider it to be sterile unless it has been contaminated with microbes from the kidney or the urinary tract. And the following are characteristics that are really important, and these characteristics typically are looked at to see if the urine being produced falls within normal limits for a number of different characteristics. The characteristics are composition, volume, pH, specific gravity, color and turbidity, and smell. The first thing is the actual composition of urine. Urine is made up 95% of water and 5% solute. These solutes consist of salts, nitrogenous waste, some hormones, some drugs, and ketone bodies. Table 24.3 lists a number of abnormal constituents that are common and found in urine. Volume. General and average urine production is up to 1 to 2 liters per day. The primary variation is going to be due to level of hydration. Factors that may impact the volume of urine produced are blood pressure, temperature, diuretics, diabetes, and the excretion of other fluids. Generally, someone needs to be producing at the minimum of 0.5 liters per day to eliminate waste from the body. Once we get below 0.40, waste will begin to accumulate within the blood. pH. Now, please keep in mind we're talking about urine pH and not blood pH. The normal range for urine pH is 4.5 to 8. Some individuals may have a more acidic diet, and that is found in individuals that consume large amounts of protein or wheat within their diet. Less acidic urine will be found in those individuals that consume diets high in fruits and vegetables. pH is also influenced by metabolism and infection. Specific gravity. This is the density of a substance compared to the density of water, and specific gravity is slightly higher than water due to solutes typically found in urine. The color of urine ranges from almost clear to dark yellow. It depends on the concentration of urobilin. With increased volume of urine, there is a lighter color. Urine that is more concentrated will have a much darker color. Smell. The smell of urine is considered urinoid, and it's the normal smell of fresh urine. Now, it may actually develop an ammonia-like smell if allowed to stand. Diabetics will actually have a urine that has a fruity smell due to the presence of ketone bodies. This is a table that actually addresses the various abnormal things that might be found in urine. Examples would be glucose, ketone bodies, protein, bile pigment, erythrocytes, hemoglobin, white blood cells, nitrites, and myoglobin. I'm not going to go through the actual conditions, but I think it is important to understand what things are considered to be abnormal if they are found in a urinalysis. Here we are looking at factors that actually influence the volume of urine. Factors that are going to decrease urine volume will be an increase in the hormone ADH, an increase in the hormone aldosterone, a decrease in the hormone ANP, a decrease in fluid intake, a decrease in blood pressure, increase in other fluid output. Examples would be sweating, vomiting, diarrhea, or hemorrhage. Factors that actually increase urine volume are going to be the opposite of those that decrease urine volume. So a decrease in the hormone ADH, a decrease in the hormone aldosterone, an increase in ANP, increases in fluid intake, increase in blood pressure, diabetes mellitus, or diuretics. Examples would be medications and alcohol. Now, micturition, this is actually the formal name for the process of expulsion of urine from the bladder. It is associated with two separate reflexes. One is known as the storage reflex, and the other is the micturition reflex. These reflexes are regulated by sympathetic and parasympathetic divisions of the autonomic nervous system, respectively, but we will also see that there is a voluntary or somatic component. Innervation of the urinary bladder and urethral sphincters. The urinary bladder has sympathetic and parasympathetic and somatic innervation. So the bladder is actually influenced by all of the electrical control systems found within the body. The sympathetic axons, they extend from T11 to L2, and these sympathetic axons will cause contraction of the internal urethral sphincter and inhibit contracture of the detrusor muscle and micturition. Parasympathetic division, this is from the micturition center within the pons. It extends from S2 to S4 in the pelvic splanchnic nerves. It is responsible for contraction of detrusor muscle, relaxation of internal urethral sphincter, and actually stimulates micturition or urination. Now, we also have to take into consideration the pudendal nerve of the somatic nervous system. This innervates the external urethral sphincter, and it contracts to prevent urination. Now, the first reflex that we want to address is the storage reflex. Now, there is continuous sympathetic stimulation happening. This is causing relaxation of the detrusor muscle to accommodate urine. It's going to stimulate contraction of the internal urethral sphincter, so urine is actually retained within the bladder. The external urethral sphincter, this is continuously stimulated by the pudendal nerve to remain contracted. Here we're basically looking at the storage reflex, and so what we want to focus on here primarily is going to be the output. We are going to have sympathetic output, and remember, this is going to be responsible for relaxation of the detrusor muscle, but contraction of the internal urethral sphincter. Also keep in mind that the pudendal nerve innervating the external urethral sphincter is making sure that that sphincter stays contracted. The micturition reflex. Once the volume of the urine in the bladder reaches 200 to 300 mils, you start to see the bladder distended. Baroreceptors are going to be activated in the bladder wall. There are visceral sensory neurons that are signaled by these baroreceptors. They will actually stimulate a center within the pons known as the micturition center. The micturition center, this alters nerve signals down the spinal cord through the pelvic splanchnic nerves. And this is primarily going to be parasympathetic stimulation. This causes the detrusor muscle to contract, also causes the internal urethral sphincter to relax. Here we can actually see what's happening here. So now we have input that is going to the micturition center as the bladder starts to fill, and then the output is actually going to the detrusor muscle. That's actually going to contract the detrusor muscle. The internal urethral sphincter will be relaxed, and the external urethral sphincter will be relaxed also. Now, what we need to keep in mind here is there is conscious control of urination, and this is initiated from the cerebral cortex through the pudendal nerve. This will cause relaxation of the external urethral sphincter, and this will be facilitated by voluntary contraction of abdominal and expiratory muscles. Once the bladder is empty, the detrusor muscle is relaxed, the neurons of micturition reflex are inactivated, and now it's primarily going to be the neurons involved in the storage reflex, they will be activated. If urination is not activated at time of first reflex, the relaxation of detrusor muscle due to stress relaxation response, the micturition reflex activated again after another 200 to 300 mils is added. Urination occurs involuntarily between volumes of 500 mils and 600 mils. It is possible to empty the bladder prior to the micturition reflex. This is accomplished through contraction of the abdominal muscles and compression of the bladder. It actually initiates the micturition reflex by stimulating stretch receptors.